Hey, hi, Suramya. Can you hear me?
Hi, nice to see you again.
Yeah, nice to see you, and thank you for accepting for this
Hi,
seminar
Alex.
webinar.
Hi.
Looking forward to it. Yeah.
Same here.
So hello everyone, and a very warm welcome to you all today's webinar. So I'm Siddhi, a part of the marketing team. So I'll be your host for this session, and I'm very thrilled to have you all here with us. So we are going to kickstart this webinar series that's on biohacking your health with a very insightful topic and an equally inspiring expert. So it's my pleasure to welcome our speaker, that's Dr. Suramya Asthana. She's an aging biologist exploring the intersection of longevity biotech, translational science, and health span in Indian population. She holds a PhD from Indian Institute of Science with 8+ years of experience in cell biology, signaling, and mechanisms of aging. She's now currently a Mazumdar-Shaw Longevity Fellow at Longevity India, IISc. She works on bridging fundamental discoveries with real-world application by fostering biomarker discoveries, therapeutic target identification, building translational pipelines, and shaping India's growing longevity ecosystem. She also serves as an editor for longevity at Research Hub and co-managing director at Nucleate India. Her mission is to accelerate discoveries that enable humans to have healthier and resilient lives. So it's a great pleasure to have you amongst us, Dr. Suramya. Over to you.
Thank you so much, Siddhi. And thank you, Fatima and Alex, for inviting me. Let me just quickly share my screen.
Yes, please.
Please let me know when you can see it.
Yeah, sure.
Okay. Good afternoon to everyone present on the call. What I'm going to do in this webinar today is to give you a flavor of the work that we're doing at Longevity India, which is an initiative that was started at IISc and now it has expanded beyond IISc to include multiple collaborators. I've been working with the initiative for the past 2 years, plus I've been working with IISc during my PhD as well. Before we start, I want to tell everyone that in this presentation, there's going to be no health or personal advice on longevity, no investment recommendations or no funding recommendations for any technologies coming up, and no personal experiences that I'll be sharing. I'm trying to give you a flavor of the research ecosystem and what it looks like in India. So this is a very famous quote, which goes by, "Everybody wants to live forever, but nobody really wants to grow old." And this has to do with the fact that there are very negative connotations that are associated with growing older. We know that the Indian population is aging rapidly, and the demographics look like they're going to hit about 347 million elderly population in India by 2050. Given that we are a huge country with a lot of diversity, it means that a lot of research is needed in this space to be able to tackle with this population that is growing. So in 2021, WHO came out with this landmark report, which talked about the decade of healthy aging, and they defined healthy aging as a combination of intrinsic capacity versus the environments that they live in, and an outcome which is functional ability. So this decade is called the decade of healthy aging, and it calls for an urgent need for developing and maintaining the functional ability of individuals and well-being as we grow older. So they've called for a lot of research, brought a lot of institutions to put in their expertise into finding out what are the mechanisms that drive aging and how we can come up with solutions for a better, healthier lifespan and health span for the human population. So we all know that aging is an inescapable reality. By definition, it is a progressive decline in physiological integrity, and this is driven by molecular damage. So here you can see that, this is what a normal trajectory of a human being looks like, where chronological age is given on the x-axis, whereas functional capacity is on the y. We know that people live for a very long time with a lot of frailty involved. And in the modern times, what we're seeing is that due to metabolic health issues, growing lifestyle habits that we have, there are signs of accelerated aging, which means people are living longer, but a large amount of the time that they live, they spend with frailty and disability. Whereas what we would want ideally is a delayed aging curve, where we spend a very short time in the frailty window, and the health span, or the time which you live in healthy conditions, is extended. So biological age is not the same as chronological age. We know that it is impacted by environments and the lifestyles that we given, but even now we have a very severe lack of the markers that can correlate biological age with chronological age. So, in the past 50 years or so, we've seen that there have been a lot of discoveries which have affected the health overall of humans, and the lifespan has also increased significantly. These have been largely attributed to access to better quality of food, better sanitation systems. A lot of research has gone into vaccination, antibiotics, building clean water and hygienic systems, as well as diagnostics has significantly improved, which has enabled us to capture diseases at early stages, maintain, a lot of discovery into therapeutics has gone in, and this has also translated into public health policies, which means that we are now living longer, we're able to live a long time and be able to manage a lot of diseases. However, we don't really know what affects health span. There have been some research that has gone into understanding what are the genetic influences which define how our health outcomes will be, as well as we know that lifestyle and environment plays a huge role in this. There are some risk factors which have come out in the recent years, which have shown that these few factors affect longevity in a huge way. So loneliness or social isolation has been attributed to the health span outcomes, as well as metabolic health is gaining a lot of traction. We know a good metabolic health is linked to longevity outcomes. Physical activity is a major player. Diets also contribute to the same thing, and sleep is emerging to be one of the factors that is very, very important for maintaining our longevity. But it becomes very important to understand where science is lacking behind and what are the unknowns that are still existing in the longevity research ecosystem. So when we all think about a disease, we can always pinpoint to a factor that is the cause of a disease, right? Be it infectious disease, we know that there is an agent causing it. But when it comes to aging itself, it's a very complex, human bodies are a very complex interconnected systems, and it makes it very hard to isolate the cause of aging. It can be a combination of multiple factors leading to the outcome, which is aging in the systemic level. We've been doing a lot of research in animal models and using that to translate that knowledge into human systems. However, we know that we've all cured cancer in mouse models, we've cured multiple diseases in worm models, but we've not been able to successfully cure the same diseases in humans because the translational gap is huge. So there's this need to build many more robust human-centric model systems to be able to study these. Then there is a lot of individual variability that contributes to aging itself, right? The genetics are different across the world. The lifestyles and environment that we are exposed to are very different, as well as the psychosocial factors that define how your health outcomes will be. For instance, if you're living in a socioeconomic background which is very different from the urban rich population, then that also defines your health outcomes. So these are some of the factors that we need to take into consideration and put them into the scientific method before we can truly figure out what are the real biomarkers that come with aging. So in 2013, a landmark paper came out which talked about aging and the hallmarks of aging. We know that aging leads to a systemic shift and the entire processes in the body change. And this has been studied from all the way to molecular level to the meta-organismal level. So the paper talks about several hallmarks of aging, and you can look at them from a molecular damage marker perspective. For example, epigenetic alterations that happens inside the cells, genomic instability or DNA damage. You have telomere shortening that was associated with senescence onset or aging itself. And you have a lot of changes in the proteins that lead to changes in the outcomes of how cells behave. This can be translated into how dysfunction onsets at an organelle level or at a cellular level, and then accumulation of these senescent cells, or the so-called aging cells in the body, leads to a lot of tissue level dysfunction that leads to organ level dysfunction, and ultimately affecting the system on a whole. These hallmarks of aging itself can be correlated in some way or the other to eroding hallmarks of health. We know that when many of these markers are off, you do see eroding hallmarks of health coming up. But one thing that has been severely lacking is that we have a lot of hallmarks of aging, but we've not been able to narrow down on a few very reliable biomarkers that will let us characterize these aging cells in the body, that will help us tell whether the individual is aging at a faster rate or the aging has slowed down or not. So it becomes essential to understand this from both a hallmarks perspective as well as how it leads to health outcomes. So in the past 9 years or so, our focus at IISc has been to study aging from a systems perspective. We use cellular models, animal models, and now we're moving on to human systems to study how changes happen in aging cells. We try to understand how DNA damage and inflammation affect the onset of senescence in cells, and then how it leads to organ level dysfunction. We try to study epigenetic changes and how they affect the aging of the human body. We do a lot of research on how proteins change in these aging cells, as well as how it affects the mitochondrial function. Mitochondrial function has been a major biomarker of aging, and a lot of research is going on into how to restore mitochondrial function and reduce the oxidative stress in these mitochondria, so that we can enable some better outcomes for the cell as well as the individuals itself. This has been my area of research, where I've been studying biomolecular oxidation and how it affects signaling outcomes. I try to understand how the molecules on the cell surface, when they get oxidized, how they affect cell-to-cell communications, and how they lead to onset of senescence in the cells. The labs also focus on understanding biomechanical alterations, glycation level changes, as well as use all of this information to build robust drug screening and delivery platforms, which we can then use to target specifically the aging cells in the body, because we know that a young cell is very different from an aged cell, and therefore the drug delivery and all targeting of drugs to these cells becomes a very different approach. So taking all of this research into consideration, we still had a lot of lacks or gaps in the knowledge in the aging ecosystem itself, which we did not know. So aging specifically, the open question in the field has been a severe biomarker shortage. There are no standardized aging markers as such. While there are multiple groups across the world that have been working on this problem, we still have not been able to come out with reliable markers that would tell us that these are the standard markers that can be used for aging all the time. The second problem is the population specificity. So while a lot of research and cohorts have been coming up in the Western countries, there have been very few cohorts in India. And we still do not know what signatures of the Western population apply to us directly, whereas what are the unique signatures in our population that correlate with health outcomes. We do not know what is the stage of the dysfunction onset. For example, we do not know whether the bone or the muscle or the brain ages first, and whether for every individual, this particular damaging organ could be the same, or could it be different? Could my liver be the weak link for me, or could it be your lungs for you? So there are a lot of individual response variations that we also need to study. And taking all of these gaps in the knowledge, then you apply it to the population itself. And as a country, we know that there is a aging population that is increasing. We have an urban-rural divide, so 70% of the population lives in rural areas. We still don't know how the biomarkers of urban versus rural populations differ. and how do their lifestyle, diet, exposure affect their aging outcomes versus the urban population, which has a significantly different lifestyle. The disease burden is transitioning in this country, so a lot of non-communicable diseases are coming up. We are the diabetic capital of the world. A lot of people with hypertension, and these numbers are surging. And growing population of aging individual means that there is a massive infrastructure deficit as well. We need a lot of care that needs to go into this ecosystem. And therefore, it calls for an urgent need to sort of build an ecosystem that would stop people from going to the hospital, whereas catch on diseases earlier and lead to better health span outcomes. So, what is the solution? I've defined a lot of problems, and the gist of it is that we know too little about aging. We know that the Western data is not equal to the Indian reality. We know that the markers might be very, very different for the Indian population. And therefore, it calls for a need for large level cohort studies in India on the Indian population, with a lot of phenotyping into continuous data coming in on genes, lifestyle, biomarkers, as well as functional capability of the individuals, which will help us build a large data sets. Now we know we're living in an era of AI, and everybody wants to use AI and harness AI to solve some of these problems in the aging ecosystem. But the AI needs data to be trained on, and if we don't have data for the Indian population, that means we don't have precision aging tools for our population. While we can use the data on the Western cohorts, we still need to refine this using our own data sets so that we can bring out more and more precision and personalized tools for our population. So the idea was to build a data set for the Indian population. And for this, we need an integrated effort. Not only researchers can do this alone, we need to bring together researchers, clinicians, different innovators from the ecosystem to work together on these problems. And this would lead to building of the entire ecosystem, which would then enable us to solve some of the problems which still exist. So this is where we started the Longevity India initiative. It is a research and disruptive technology hub to enable healthy aging in India. The hub is in Indian Institute of Science. A few professors from IISc came together and we formed the India Longevity India initiative. It started off as a virtual hub, but we are now on to building our own lab in IISc, where we can serve as a data repository, a biobank, as well as doing a lot of research which is centered around aging. And I will talk a little bit about what we do. So one of the moonshot projects that we have taken up is called the Bharat Study. The Bharat Study stands for the biomarkers of healthy aging, resilience, adversity, and transitions. I'll talk about more in detail about the Bharat Study in the coming slides, but we also do a lot of other research under the Longevity India banner. We build organ systems that will help us to understand what are organ-specific markers of aging. We try to use these organoid models to understand what can be potential therapeutic targets, and to also use these systems to screen for drugs that will work for anti-aging therapies or not. In the Indian ecosystem, we have a lot of traditional knowledge. So in the past few years, we've been working with the Ayush Ministry, as well as a lot of institutes that work on Ayurveda, that have a lot of databases on traditional Indian knowledge. And we want to bring a lot of evidence into this traditional knowledge so that we know that what has been used in practice in the Indian ecosystem actually works or does not work, and to add evidence to those practices. And at last, the ecosystem itself needs a lot of awareness and education. And for that, we have a huge outreach arm. We have campus ambassadors, which are students that are studying in different universities across India, and they help us spread awareness, hold webinars, seminars, and different initiatives on their campuses so that we can spread the word and encourage more people to take up problems in the aging research space. We have a podcast where we try to bring up different themes and topics around longevity, and specifically from an Indian context, so that people know what is it that is the latest in terms of the research. We have a monthly newsletter which also aims to feature a lot of technologies coming up, a lot of research, and also feature the people that are working in the field. And we held the first Longevity India conference in March of this year, which was called Rise for Healthy Aging. We had a lot of participation from academia, industry, policymakers who attended this meeting. And the hub itself is a collaborative effort. So we're open to collaboration, which brings together people from across all four fields of science, medicine, technology, as well as society. So coming to the Bharat study, this study was taken up mainly to define what is the baseline in the Indian population of aging trajectories. So we did not know what markers come up with age, whether these markers are leading to poorer health outcomes or better health outcomes. And so we thought that it is necessary to collect data from individuals across the spectrum, and which is why we include healthy individuals aged 18 all the way up to centenarians across different strata. So we try to maintain urban plus rural populations, and we maintain a gender balance also in our cohort so that we get a rich data set which is truly representative of our population. We measure, we do a deep phenotyping of what are the functional changes. This is done through a metadata that is collected through our clinicians and clinical partners. We try to assess what are the cognitive function in these individuals, metabolic alterations, epigenetic changes, gut microbiome changes, biophysical changes in the cells and different tissues that we take, as well as a deep immune phenotyping of the individuals. So all of these samples which are collected is used to build a multi-omics profile, and we do a proteome, epigenome, metabolome, and lipidome analysis from each individual, which will lead to building of the first Indian age map and also give us a biological age reference for the Indian population. And it will also help us build out population level trajectories and see which of the biomarkers and features come up with aging and which can be used to build novel therapeutic targets. So going into a little bit of detail on what we do collect, we collect these 5 biological specimens and they're used for different types of analysis. You can see the details that we go to. Mostly the study is centered around multi-omics data because multi-omics gives you a very rich data set that is untargeted, unbiased, and we're not using the same information that is come out of the Western population, whereas trying to find novel biomarkers as well. This biological data is complemented with a huge metadata that does functional recordings, be it vitals, cognitive assessment, frailty assessment, as well as the socioeconomic background, their lifestyle habits, and the kind of food they consume. Ultimately, we're going to use this to build an integrated AIML pipeline for biomarker discovery, and this will lead to building healthy aging outcomes as well as organ-specific clocks for the Indian population. We've been supported by the Indian Council for Medical Research, and we're a Center for Advanced Research in Aging at IISc, which is enabling us to carry out this large cohort study. But now we know that various systems in the body age at very different rates. It can be possible that every organ in the body is ticking at a different rate, and every organ can have its own organ-specific clock, which is the reason why we wanted to complement the Bharat study with some organ-level data, because naturally any human participating in the study is not going to give me their organs to be able to analyze. So we thought we'll develop some organ systems in our labs and use them for cytoprotective drug screening. So these are the different models that we've now developed over the last few years. We develop lungs and livers. We work with blood and body linings where we try to understand how epithelial cells, mesothelial cells are aging. A map of the endocrine and exocrine systems in the body, as well as skin, which is the first layer of contact with the exposome. And all these model systems help us understand what are the unique features that come out of each of these organs and help us identify the organ-specific markers. So these signatures will then be correlated with the Bharat study data that we have and help us define some of the organ-specific clocks. Now we know that at a cellular level, multiple aging cells or senescent cells accumulate in the body. And over the last few years, the senolytics or drugs that eliminate these senescent cells specifically in the body can lead to better health outcomes and are popular to be used as anti-aging therapy. These could be diet, supplements, various practices, or peptides and drugs that are now coming out. So we use our model systems that we've developed in the lab for anti-aging drug screening for cytoprotective as well as radioprotective screening. And we're also working with ISRO to understand how can these model systems be used to study the effects of radiation and microgravity as well, because we know that humans are going to be going to space in some time, and so we need to understand how aging occurs in space and how can we protect our astronauts better in terms of their health outcomes. So we know that there are a lot of drugs out in the market, all the way from some supplements to snake oil, and we don't know whether they actually work or not. So we are using our model systems to test out all of these drugs, see if they work for all of the organ systems or whether they are specific to a particular system so that they can be used in targeted therapies. And at the end, we know that exposomes or environmental factors drive aging. And these are the ones that can harm a particular organ in the body more compared to the other. And so there are a lot of unknowns also in the field. This means that the environment itself that we live in make it very make ourselves very unique to the type of aging outcomes that we have. And so we don't know how that affects gut aging. So we're trying to understand in a country like India, where in every 80 kilometers your microbiome will change, how to find out these unique features of the gut microbiome, how to see that the lifestyle practices or the food that is consumed in local regions will affect the microbiome diversity and the health outcomes. How does it affect the epigenetic aging, which is the first layer that gets affected in response to the environment, as well as what outcomes does it have on hormones and hormonal aging. We don't know how all of these exposome factors will lead to supplement specificity and ultimately use all of this information to build some precision therapies. So, all in all, I wanted to close with introducing the team itself. We have a huge team, which is not only professors from biological sciences, but we have people from diverse backgrounds, including AI/ML experts. We have people who have been working historically in developing 3D model systems, in understanding systems biology, as well as developing nanotechnology solutions, which can all be utilized towards the aging research questions that we have. And we have a team of clinical collaborators as well, which are from hospitals in and around Bangalore right now. And we're speaking to a lot of partners that help us do our sampling and help us evaluate the clinical technologies better. So together we form the team of the Longevity India ecosystem. But this doesn't come without the partners that we have. We work with academic institutions so that we can build better collaborations and see how we can complement each other's skills. So we have collaborations with IIIT Delhi, JSS, as well as INSTEM. And a lot of clinical partners are involved because the clinicians become important in serving as like the first layer of contact with the individuals that are volunteering to participate in the study. So all of our clinical collaborators work with us to do the community outreach, to identify the volunteers that will partner in the study, and also given their expertise into recruiting the people who will be part of the study, declaring whether they are healthy individuals or not, and also using this data that is coming out of the Bharat study to ultimately apply it back into clinical practice. We know that for the first time, industry is at a stage where it has made a lot of innovations, and therefore industry partnerships are very important for us to be able to do the research that we are carrying out. So we've partnered with a lot of diagnostic labs, including 1mg, Healthians, and Lalpath labs, and they serve as the diagnostic partners for us to be able to do the existing clinical tests that we do in all of our cohort studies, as well as companies like Beckman and Bruker have come on very early at a very early stage with us to give us access to their technologies, so that we can add more and more depth and rigor to the studies that we are doing. We're working with companies like Arca to develop in-house solutions and platforms that will help us capture metadata better. We work with a company called Diagnostics, and they've developed an age reader that helps take the fluorescence reading from your skin and understand how advanced glycation end products are accumulating in your body. So we're trying to see how their technology applies into the Indian population on Indian skin tones, and then ultimately use the same data sets and the same technology, but standardize it for the India ecosystem. So we also work with companies like and a lot of longevity clinics like Biopeak, and this helps us bring in access to a lot of community that they are developing, because they see people who are enthusiastic about their health, and so they can also be included into the Bharat study. Decode Age is helping us build out the microbiome pipeline that we are doing, and the community partners that we have, including Biovikas, AHPI, as well as Dementia India Alliance. These are the organizations that are very society facing, and they're building communities of senior citizens or individuals that are into their fold. And we partner with them to get access to the communities, as well as do a lot of these health check camps where we can recruit people into the Bharat study, as well as give back information to the people in collaboration with our clinical partners to be able to help them better. We work with AHPI, which is access to about 7,000 hospital partners in the country. And this helps us spread the Bharat study from Bangalore all across India. And we're also speaking to other collaborators now to take the Bharat study from Bangalore all the way to different cities. And with that, I'll come to the vision that we have for a healthy India. So while we're doing a lot of innovation and discoveries at a cellular level, and which will lead to biomarker discovery and therapeutic target discovery, what we ultimately want to do is build some of the Bharat standards that we have. This comes from a huge gap that exists in the clinical ecosystem, where we are still using Western population data to define the clinical benchmarking for our population. But ICMR itself has taken on some efforts, so that we're able to redefine what the clinical parameters or reference standards look like for India. And at the intersection of that lies the societal impact that we're going to have. So ultimately, what our major goal is to be able to come up with biomarkers that will add knowledge in a way that will reduce the rate of hospitalizations of older people in the population, because we aim to enable health span outcomes and add some dignity in the way people are aging. A lot of preventive health outcomes will come out of this, and a lot of improved quality of life. And this is why it becomes important for a huge collaborative effort like this to come together, and for people to participate more and more into the ecosystem. So I'll stop here, and I'll move the floor open to some questions now, so that we can have a more interactive discussion and answer any specific questions that you have.
Yes, thank you so much, Dr. Suramya, for that insightful and thought-provoking talk. As you mentioned, we'll open the floor for quick questions. So please drop in your questions in the chat box so that we can read it out to the speaker.
I just had a quick question, right? So, what is the outcome that you have already attained based on these studies, and how is the data looking different, and what are the interesting things that you have spotted based on the studies that you have done so far?
Yeah. So I'll answer from both the Bharat study perspective and the organ aging model. In the Bharat study, we've done a deep immunophenotyping, and we see that some of the markers that are coming up are different from what we saw and what has already been reported in the Western population. And these results were also presented at the conference that we have. These are very early data sets, very small population, a subset of the entire cohort that we have. But we do have some inflammation markers that are very different from the reported cohorts. The main reason could be that we live in a tropical country and we have a lot of infectious diseases. So a lot of Western population likes to call us chronically inflamed Indians, but that may not be true for us. We may have different markers, different features that are coming up. So immune data suggests that there are some different features. And the second one, which is a very recent finding, but again, a subset of the cohort. So we do see epigenetic markers, which are very different from what is already known in your standard clocks. So the clocks that are classically used for biological age calculation, they don't work very well in our population data cohort. So they're very different, which is coming out of the Bharat study. Again, the numbers are too small at this moment to make any claims. So we're adding more and more research and more numbers to the cohort to answer for that. The second one is from an organ aging perspective. We've been successful in developing a lot of aging model systems for different organs. And we have leads from a few drugs that are already approved. They're selling in the market as, popular supplements. And they do show benefits across all of the model systems that we have. So we're now looking at collaborative projects which will take up these studies in human trials, because these are supplements that are already being consumed. So once the human trials go out, only then you can say that the results that you saw in the cell studies are directly applicable. So we do see some interesting drugs coming up. A lot of drug repurposing is also done in the labs. So existing FDA approved drugs we're now taking up to do some new human level trials.
Yeah, one last question before I go. I just wanted to know the data sets that you're building. At the end of the study, is it going to be publicly accessible?
Yes, so we're an ICMR Center for Advanced Research, and by ICMR guidelines, all of the data for this cohort will become public via the ICMR repository.
Okay. Thank you. Thank you, Suramya. It was a great talk. I absolutely enjoyed hearing you.
Okay, so we have one question. We have two questions in the chat box. So the first one is from Jose. When cell surface molecules get oxidized, how does that affect communication between cells, and how does this play a role in the aging process?
Yeah, great question. And this has been my work for the past 8 years, where I try to understand how the lipid bilayer, which is there on the cells, if you have lipid peroxidation, or if you have oxidation in the cholesterol molecules itself, what it does is it interferes with the protein structure. So these molecules can then go and hinder the protein structure itself. It will not let the receptors on the cell surface activate and communicate in the way that they want to. So this is something that I saw with a GPCR that I was working with. And this GPCR essentially is dependent on cholesterol for dimerization on the cell surface. So dimerization and oligomerization both. So, but if you had an oxidized lipid environment around it, then the receptor itself would not function. So if you have an extracellular cue coming in, then the receptor would not get activated and it would not relay the signal inside, which means that normally the receptor is involved in cell proliferation, in migration of the cells. But when you're not able to relay the information inside, that means the cellular behavior itself alters. So we saw that the inflammatory axis was off, the migration of the cell was not as it was supposed to be, and the cells were obviously not proliferating. So this means that, and when you quench some of these molecules from the cells, which are oxidizing the lipid environment, then you're able to see a lot of restoration in cell communication. And this was one receptor that we studied, but it could be an effect that could be applicable to other receptors as well. In the aging process itself, how does it play a role? We still don't know whether it is a cause or an effect, but when you alter the cell-to-cell communication as well as how the cell behaves, that leads to a lot of cellular dysfunction and accumulation of these damaged cells in the organ, and that ultimately leads to the tissue level dysfunction.
Thank you, Dr. Suramya. We have one more question from Akshaya. Okay. Okay, Jose is asking a follow-up question. So this damage can be prevented and age reversing can be accelerated.
The damage can be prevented. However, at a cellular level, you can look at it as an isolated event. So in cells, you can quench, let's say, a lot of oxidative stress from the cells, and you will see cell communication coming back. But whether that translates to an organ level aging phenotype, we still don't know yet. We have an ongoing experiment where we're trying to use a drug targeting these molecules and seeing whether it has beneficial output. So as soon as that comes out, I'll let you know.
Thank you. We have one more question from Akshaya. She's asking, what is the sample size of the Bharat study? And also cell-free and DNA quantification from the biofluids are planned, and she wants some insights on the same.
Okay. Yeah. So I'm sorry I missed that in my talk. We have designed the Bharat study in a way where we partner with clinical collaborators, and each clinical center gets us ethics approval for 1,000 individuals per center. So currently we are approved for 5,000 samples in urban and rural Bangalore across 4 different centers. And we're speaking to 3 other collaborators that will help us take this study to Chennai, Delhi, and Bhubaneswar, and each center again gets 1,000 individuals added. So that is it's an emerging landscape of the overall sample size, but hopefully in the next 5 to 10 years we'll be hitting 10,000 in numbers. Next part was, I think, the cell-free DNA. Yeah, so we are that was not in the initial plan, but because of the recent interest that has gone into understanding cell-free DNA, even microRNAs, long non-coding RNAs, and how they correlate with aging outcomes, we are thinking of including a little bit of profiling of these miRNAs, long non-coding RNAs, from the plasma samples itself. Uh, in terms of other cell-free DNA markers, we don't have a very defined plan around that yet, but we're happy to look into that and include it.
All right, thank you. So we have one question that was filled in the form. So that is, how can we lay a framework for India towards longevity to add to our economic strength?
Right. So I think this is part of it. I also covered in my talk, where we're trying to develop the Longevity India ecosystem not only as a research technology hub, but we're working with policymakers very early on, so that whatever findings we have can be then translated into outcomes as well. And this is where we are now looking at forming an association for healthy aging. This is a collaborative effort where Longevity India is one of the initiatives that are participating, but we also have initiatives like AHPI, Biovikas, Dementia India Alliance, which are already working at a national framework level to influence some of the policies that are around these healthy aging outcomes. And we are trying to bring in policymakers, because aging itself is not only about the biological age. It comes with a lot of economic implications, financial implications. You have to plan for your years ahead, and we all know people are not really retiring at 60 anymore. People are pushing the age to which they're at which they are still working. So we need a lot of changes in the entire framework. While Longevity India has started as the research framework right now, we're slowly bringing in more and more people, so that it just does not remain as a research project that we took on. But ultimately what we want to see is that research is done at Longevity India. It gets translated and it gets to the public via the Indian Council of Medical Research, or DST, DBT, the bodies that are there, which help us translate the scientific knowledge better, and then use all of this into the clinical practices. And having ICMR in the fold helps us enable this a little bit, because early on, if you bring in clinicians, if you bring in the industry, if you bring in everybody who's working on the entire project together, then you are able to make changes a lot faster. So a small example for this is that Biovikas had been working with at the ground level with a lot of us, and they were able to influence a certain level of policy decisions that were made recently about the elders and their health insurance. So if you bring in people like this from very early on and have them included in the aging research that we are doing, then ultimately it leads to a lot of beneficial outcomes that you see. In terms of economic strength, I think the more startups that come out, the more people who take up these projects, the more people who use the data sets that are very India specific, and then translate this into therapies, developing startups that target really the mechanisms of aging and the biology of aging, that will lead to a lot of meaningful impact and outcome that will help to grow our economic strength better.
Right, thank you. Uh, does anybody else have any questions? All right. Looks like no more questions. Okay. If there are any more questions, we'll maybe email it to you and you can clarify it for us. So thank you so much, Dr. Suramya Asana. It was a great, it was, thank you so much for sharing such valuable perspectives with us today. So it was great to listen to your talk as well. So thank you, everyone. Thank you, everyone, for your questions and engagement. And it's been a great discussion so far. And on behalf of the team Azuka as well, I would like to extend my special thanks to you again, Dr. Suramya. Thank you so much for joining us today. Thank you.
Thank you. Thank you so much.
Thank you. Okay. So regarding the next event, regarding the next webinar, we'll be posting the time and about the details regarding the speaker on our LinkedIn and Instagram pages. So thank you.
Thank you, Zuka.